change is Will Stone is going to present on my behalf. I think often in these symposia you have speakers who have Y chromosomes and short telomeres, and I think it is good to actually allow the slightly younger people to present. Will is the first presenter. He did his PhD in Radboud University Medical Center, and I moved to the London School, where he continues to work on naturally acquired transmission blocking immunity.
Thank you, Teun, and good morning, everybody. I'm going to talk about natural and vaccine-induced immunity against novel P-falciparum gamete targets. So as long ago as the 1950s, it was shown that human immune responses could affect the likelihood that mosquitoes feeding on a person would become infected by the gametocytes in their blood. However, it wasn't really until the 1970s that the concepts of transmission-blocking immunity and transmission-blocking vaccine development were cemented by these two major studies by Robert Gwadz, Richard Carter, and Florence McAuliffe at NIH. And what they did is they inoculated chickens with formalin-treated gametocytes and gametes, the transmission stages of the malaria parasite, for P. gallinaceum. And what they noted is that chickens who were inoculated with a larger number of gametes or gametocytes infected mosquitoes with less oocysts. So these chickens were developing antibodies in response to the inoculum that would interrupt transmission in the mosquito mid-gut. They also identified two proteins, P230 and P4845, that were thought at this time to be the key antigens response, the key targets of these transmission-blocking antibodies. And subsequently, it was shown that both of these proteins function integrally in gamete binding and fertility. Now, in the decades that followed these studies, numerous others showed that humans also develop transmission-blocking antibodies. And this is because, though gametocytes are generally protected from antibodies inside the red blood cell, when they break down, antigens on their surface are exposed to the immune system, and they elicit antibodies. These can be taken up by mosquitoes when they feed alongside viable gametocytes. The antibodies then interact with proteins on the gamete surface that are shared by gametocytes, and they block transmission. In humans, the presence of anti-PFS230 and anti-PFS4845 antibodies in Plasmodium falciparum is strongly associated with the sera as a whole having transmission-blocking activity. But around 30% of individuals with transmission-blocking sera lack antibodies to both 230 and 4845. So it appears like responses to these two proteins are not answering the whole picture. Some other antigens may be responsible as well, So the way that we test the transmission-blocking activity of sera or antibodies is we perform the standard membrane feeding assay, the SMFA, where we add test serum or antibodies to infective gametocytes from culture and compare their effect on mosquito infection rate, read by the number of oocysts in the mosquito gut or by luminescence if you're using transgenic parasites, compared to a control that doesn't contain the test serum or antibodies. And that gives you a metric which we call transmission-reducing activity or percent inhibition. So to go back to PFS 4845 and 230 antibodies, to try to dig deeper into what their contribution is to transmission-reducing immunity in humans, we took individuals for which we had large volumes of sera who blocked transmission in the standard membrane feeding assay. We purified out all of their IgGs and we tested them in the SMFA. And as you can see here, I've got a laser. No. As you can see, individuals A, B, and C all blocked transmission to mosquitoes. We then purified out from their total IgG only their PFS 4845 IgG. And one individual possessed antibodies against this protein at physiological concentration that was able to block transmission to mosquitoes. When concentrated 9 times, a further individual was turned into a blocker from a non-blocker, indicating that they had antibodies, but they weren't at sufficient blocking concentration in humans. Without their 4845 IgG, all 3 individuals' total IgG continued to block transmission. Removing the 230 IgG and testing that independently showed that at physiological concentration, these antibodies did not block, they only mildly reduced. but concentrating 9 times again, and 2 individuals blocked transmission. And this may not seem shocking. 230 and 4845 definitely induce transmission-blocking antibodies, but this is the first demonstration that antibodies elicited by natural malaria infection specific to these proteins actually causes transmission-blocking immunity. So it's something we knew, but we've confirmed. Importantly, without 4845 and 230 IgG, and in the presence or absence of complement, and 230 activity is dependent on complement, these sera continue to block transmission. So this indicates that other antigens are definitely inducing transmission-blocking antibodies. On top of this, we performed surface immunofluorescence assays with activated Plasmodium falciparum NF54 gametes that failed to express PFS 4845 or 230 on their surface because they were knockout parasites. We'd developed this line in our lab. As you can see in the top right-hand corner, there we go. Monoclonal 4845 and 230 antibodies did not bind proteins on their surface, so the proteins were not present. However, transmission-blocking sera depleted of 230 and 4845 antibodies, or not, did recognize gamete surface proteins. So this confirms that they are recognizing other antigens on the surface. So immune sera can block transmission without dominant PFS 4845 and 230 antibodies and recognize other antigens on the gamete surface. To try to work out what these might be, we performed the standard membrane feeding assay using the purified total IgG of 648 individuals who were malaria exposed, mainly from West Africa, but also some highly exposed Dutch expatriates. We identified 22 individuals who blocked transmission reproducibly in the standard membrane feeding assay. That's more than 90% inhibition of osis density. To work out what antibodies might be responsible for this, we developed a gametocyte-enriched protein microarray. This was comprised of more than 315 primarily gametocyte proteins, and our aim was to probe all of these sera on that microarray as well and compare the difference in reactivity to these different antigens between the blockers and the non-blockers. And to create a worthwhile comparison, we only compared the blockers to individuals who were gametocyte exposed, as assessed by microscopy, and who had less than 10% transmission-reducing activity. So we identified 43 novel gametocyte antibody responses that were associated with this high level of transmission-blocking activity. You can see here 4845 and 230 responses are higher in blocking sera, and the same is true for the 528 peptides present on the microarray, representing 315 proteins. However, on an individual level, when we performed an empirical Bayes t-test, the response of proteins among the blocking group was significantly higher than the non-blocking group for 43 proteins. When we took the sera from these individuals, and this is a time span of about 15 years where we collected these samples, we also performed direct membrane feeding assays to get an idea of the person's actual infectivity. So that it represents the contribution of their gametocyte density, maturity, sex ratio, but also their immune response. What you can see here, sorry, I'm useless at this. The different colors represent different antigens. So yellow is PFS 4845, blue is PFS 230, and the individuals in the right-hand column of all of these are seropositive individuals. So individuals who were seropositive to PFS 4845 and 230 infected significantly less people in the field. The same is true for the novel gametocyte proteins. So individuals who were responsive to significantly more of the novel TRA biomarkers infected less mosquitoes in the field and less people infected sensu strictly. The same was true for gametocyte proteins that were more likely to be surface expressed. So, naturally acquired PFS 4845 and PFS 230 antibodies can inhibit transmission, but do not fully explain the phenomenon. And antibody biomarkers of TRA, identified from 22 blocking individuals in the standard membrane feeding assay, predicted lower infectivity to mosquitoes among 366 individuals in the field. This is all very interesting, and biomarkers of transmission-reducing immunity are, of course, very important and interesting epidemiologically. But it's more interesting if any of these biomarkers actually translate to novel transmission blocking vaccine candidates. We at this stage don't know if any of them actually cause TRA. Didn't know if any of them actually cause TRA. Spoiler. So to work out whether any of these do elicit functional transmission blocking responses, we have been working with MVI to produce the 43 novel TRA-associated proteins in the wheat germ cell-free system, along with cell-free sciences. This attempt yielded 22 proteins at reasonable enough concentrations to proceed to immunization studies. Our immunization protocol was 3 bi-weekly doses of 20 micrograms of the wheat germ cell-free antigen in a Montanide ISA-720 adjuvant, and we used an R06C, which is based on a subdomain of PFS 4845, as a positive control. We bled the mice at day 42 for the first time, and purified out the polyclonal antibodies and tested these in the standard membrane feeding assay and in various serological assays. Of the first batch of proteins, first batch of 13 of the 22 proteins that we have, 11 out of 13 showed some level of precipitation, and in some cases that was quite extreme. So the wheat germ cell-free system with these proteins is kind of reflecting our history of attempting to produce gametocyte proteins in that it's notoriously difficult. However, all were immunogenic in mice, and 6 candidates of the 13 recognized gametocyte proteins in fixed, soluble, permeabilized gametocytes in surface immunofluorescence assays. Immune sera from 4 of the novel candidates show reactivity with actual activated gamete surface proteins, including gamete egress, sporozoic traversal protein, and surprisingly, the most reactive protein, heat shock protein 4T type 2. In the standard membrane feeding assay, only 2 of the 13 showed reproducible transmission reducing activity. These were PF11.1 and, slightly shockingly, liver stage antigen 3. PF11.1 is a well-known protein. It's a megadalton protein, very, very big, and it's expressed by the gametocyte and is involved in their egress from the red blood cell. You can see from the gametocyte cephas at the top that it localizes in the parasitophorus vacuole and is involved in gamete egress during activation in the midgut of the mosquito. Liver stage antigen 3, on the other hand, was only included on the array because it was identified as a protein that is transcribed, or rather expressed, during early gametocytogenesis, but only at a very low level. It's much more enriched in asexual stages. So we were a bit shocked by this finding. However, we have tested some sera from immunization studies at Radboud, where LSA3 was used as a potential vaccine candidate. We selected the highest titer sera, tested these in the SMFA, and it did not confirm the finding here. So we are not getting overexcited that we've stumbled upon a new vaccine candidate. We think this is really unique to the particular peptide that we produced in the system here. We will need to confirm this. So work that is ongoing. There are 9 more proteins in the wheat germ system to proceed to immunization studies, although only 5 are soluble, so only those will be tested. 13 antigens, those listed in this table here, will be prioritized for production in a Drosophila S2 system, in the hope that that will be more successful than the wheat germ. And these are prioritized because of the likelihood of their being expressed at the gamete surface. So they all possess either a signal peptide or a transmembrane domain, or they've been empirically shown to be present on the gamete surface. In addition to this, we're hoping to produce a bigger, better microarray based on all available gametocyte proteomic data sources, rather than just one, as the microarray in this study was based on, which we hope to then employ with samples more globally than just West Africa. Brings me to a close. I will mention Roos de Jong and Matthijs Joor at Radboud, who have been involved in the immunization and protein production aspect of this work more than myself. And Teun Bousema, of course, my PhD supervisor. Thank you.
All the 3.
Hi, very nice
work. Thank you very much. I had one question. When you're saying that you depleted PFS230 from your population, do you have how are you doing that?
Yeah, so we didn't. It's likely that we didn't deplete all 230 antigens. So the construct that we were using on the affinity chromatography columns was the 230 CMB Fraunhofer candidate. So that's amino acids 444 to 730, which is the immunodominant transmission block. Most transmission blockage comes from that segment, but there are 13 other cysteine motifs in PFS230 that we wouldn't have been depleting the antibodies from. So the main evidence for 230 is that we know that's the dominant transmission blocking epitope contained in that protein, and transmission reducing activity was independent of the presence of complement.
Yeah, but there's a lot more 230, and many of the monoclonals don't see region C, right? That are transmission blocking, 1B3 and stuff. So
right.
Be a little.
Hi, we're Takatsubo from Japan.
Hi.
And first question is the same as Kim. The domain C is very important, which you showed completely independently. That's my happiness. This is a comment. And secondly, the question is the LSA3. So now I just missed some explanation that anti-LSA3 antibody which you produced is recognized gamma site in IFA. Anti-LSA3 antibody recognized gamma site.
No, LSA3 didn't recognize gametocytes in the CIFAs that we performed, and it had very limited recognition of gametocyte antigen in a lysis ELISA. The appearance of transmission-reducing activity in the SMFA is as inexplicable to us as it is to you. Yeah.
Okay, so that means you don't know the reason why that's blocked.
No.
Okay, I'm expecting to solve that problem.
Thank you very much, Michael Good from Australia. In looking for other targets for antibodies, I wonder whether you might have also been looking for other types of immunity. I mean, many years ago, Carmi Mendes and Richard Carter demonstrated that cytokines working TNF, working through an arginine system and nitric oxide and so-called crisis factors, could completely block transmission.
Yeah.
And that was obviously not an antibody-mediated form of immunity, but it was something that was engendered by other proteins in the gametocyte, stimulating T cells, essentially.
Yep.
And Hart and Target have proven that previously in a mouse model. I wonder whether you consider looking for T cell targets of transmission blocking.
It's certainly a valuable approach going forward. We are interested in inflammatory markers and how they relate to transmission-reducing immunity. In this case, we were able to exclude that as a source of this activity because we were purifying IgG, so what we were seeing was specifically related to the antibody using correct filter sizes and
no one's saying the antibodies can't block transmission, but I'm saying that there are other mechanisms which can be as important, if not more important.
It's definitely true, yes. Splenectinized monkeys in the early 90s proved that during crisis, cytokines can directly kill gametocytes. So it's an avenue to look down, yeah.
Yes, I just wanted to make a comment that I think, at least maybe I'm biased in my thinking, but in our experience, the parasites seem to have very immunogenic epitopes that produce a lot of antibodies and they do not have transmission blocking. So, for example, in our case, our best transmission blocking monoclonal does not recognize the gametes. And we think this is because the protein suffers a conformational change and only exposes this temporarily. So just keep this in mind. I would say do not rule out the ones that are SMFA negative. Go for the activity assay, the functional assays, because there are surprises. Nothing we predicted worked the way we thought it would work in our case.
Yeah, yeah.